Hello, my name is Blair, and I'd like to thank you for taking the time to look at my post today. If you need to, feel free to pause this video at any time, or you can always download the PDF. We have previously described sustained changes in cytokine expression and T-cell profile in burns patients more than 3 years after injury. This is interesting to us because we know that burn survivors are at risk of a range of morbidities, and we'd like to be able to track the immune profile to clinical outcomes. We don't actually know what the trajectory of the immune profile looks like from the acute injury to the long-term displacement. So what we have done is we have designed and optimized two complementary panels profiling CD4 and CD8 T cells for a 5-laser Fortessa. The panels that we've designed, they actually share a base of 12 markers, including the immunosuppressive marker PD-1, which was something that we had not previously investigated in our other study. The CD4 panel allows us to track changes in regulatory T cells, as well as a range of the most common T helper cells, while the CD8 panel focuses more on markers of cell activation. So we've got no affiliation with Fluorofinder, but I did just want to say that I have found their tools the most useful for pre-optimizing panels before any wet work is done, and it's particularly assisted with ensuring co-express markers were spectrally separated and bright fluorophores were matched to markers with low expression. So we optimized our panel using serial two-fold dilutions, with each cocktail used to stain 1 million cells in 96-well plates. The optimal concentrations were determined using the stain index calculation, which takes into account the comparative brightness of positive and negative staining populations and the spread of those populations. The table on the right shows the optimal dilutions as we calculated them. To account for interrun variability as we use this panel, with samples that are accumulating in our biobank, we've actually stored a cache of PBMCs from a control donor to be used each time the panel is run so that we can normalize that data between experiments. Some key points to note in our gating strategy is that we use a logical AND gate to identify CD25 and FOXP3 positive Tregs that are low in CD127. We use CD45RA and CCR7 to define our memory subsets, and we use bivariate gating of CXCR3 and CCR6 followed by CCR4 to define our T helper subsets. And as an added bonus, I'm including some data here from a study that was conducted by our honours student Harry, with some help from our new favourite statistician Gemma Cadby. What this panel has shown us so far is that immune profile, when blood is first collected during the first revision surgery, which is usually within 5 days of the original injury, that actually correlates with outcomes measured at later time points. And going forward, what we want to do is we will use this panel to track the trajectory of T cells in these patients as they return for repeat sampling at months to years after the injury. Thank you for taking the time to review our poster, and if you have any questions, feel free to get in contact.